All right, it's my pleasure to introduce Divya Rose. She's a PhD student with Dr. Matt Griffin and Dr. Graham Rosser at Mississippi State University's College of Veterinary Medicine. She's been working with fish in some capacity for the last 12 years, starting with her bachelor's in fishery science from India and continuing with her master's degree, which she earned at Mississippi State University in the College of Forest Resources, Department of Wildlife, Fisheries, and Aquaculture. Her area of interests are in molecular bacteriology and its applications to fish health. Looking forward to hearing your talk.
Thank you, Dr. Phelps. I'm Divya Rose, graduate student from Mississippi State University, and today I'll be talking about part of my objective, searching for environmental reservoirs of antibiotic resistance in commercial catfish ponds in Mississippi Delta. So pathogens, bacterial pathogens adapt over time and acquire drug resistance. And if you think about it, antibiotics are naturally present in bacteria even before it was being mass produced by human beings for treatment. And they have the natural ability to resist these antibiotics. But use of antibiotics over time have resulted in propagation of antimicrobial resistance between different microbial communities. And the main vehicle for this propagation are plasmids, which are extra chromosomal genetic material which have their own replication mechanism and carry genes for antibiotic resistance, mobilization, and conjugation. When it comes to catfish aquaculture and antimicrobial resistance, the three approved antibiotics for use in food fish are aquaflor, romit, and terramycin. And over the last 2 decades, we've been seeing an increase in the number of antibiotic-resistant isolates that is being reported at the Aquatic Research and Diagnostic Laboratory here. This graph can be misleading because fluorphenicol was only approved for use starting from 2007, and this drug was not tested for before this time. But still, we are seeing an increase in the number of antibiotic-resistant isolates. And throughout this presentation, I'll be talking about Edwardsia chloride a lot, which is a major pathogen of concern for channel catfish fingerlings, which causes enteric septicemia of catfish characterized by the classical hole in the head lesions. We've had multiple records of multi-drug resistant isolates of Edwardsia chloride over the last 30 years. And the one main reason I'm working with the pathogen is it's heavily studied. We know how the pathogen works, and we have an established challenge model. So with all that, my objectives were to characterize multi-drug resistant plasmids in archived E. chloride isolates from 1994 and identify the antibiotic resistant gene reservoirs and associated mobile genetic elements in catfish aquaculture systems. So first, we worked with the historic antibiotic-resistant E. chloride isolates. We had archived isolates that were resistant to oxytetracycline and romit, and we sequenced them using both nanopore and Illumina technology. And when we did a MOV alignment, we saw a region of non-congruence in the multi-drug resistant plasmid that was present in the historic isolate. So we did a sequence comparison. The sequences, the multi-drug resistant plasmids were showing a 96% sequence similarity with an area of disagreement. And if we zoom into it, we can see that the historic isolate lacked the gene cassette that confer resistance to fluorphenicol. So this kind of evidence towards the fact that plasmid machinery to acquire these antibiotic resistance were present even before the drug was introduced into the industry. Another interesting fact we saw associated with this historic isolate was the presence of these huge plasmids with two origin of replications. So usually the EAC is described to have two native plasmids of the size 4.8 kb and 5.6 kb, but we started seeing these huge plasmids with multiple origin of replication of 16 kb and 10 kb. Initially, we thought this to be a sequencing artifact, but 6 out of the 8 sequenced historic isolates had these big plasmid. So we extracted the plasmid and tried running a gel just to make sure we do not have any, big plasmids there. And as you can see from the image, all the plasmids that we had lied between the 4 kb and 6 kb molecular weight markers. So then we looked at the raw sequencing reads and mapped it to the NCBI reference sequence of this plasmid. And as you can see, all the highlighted bases are the disagreements to the reference sequence. And we had enough read support for the regions of disagreement. And this was consistently found across multiple historic E. telluri isolates. And we suspect this to be a case of heteroplasmy. So heteroplasmy is a situation basically in which multiple alleles of the same plasmid coexist in a bacterial cell. And over time, as a part of genetic drift, one of the alleles gets established in a community. So as of now, why heteroplasmy is present in this historic isolate is unknown, but recent studies suggest that the E. telluri has evolved to maintain just one copy of these 5.6 kb plasmids. We also found a phenotypic difference. There was a reduced growth in the antibiotic-resistant isolates, as you can see from this OD in overnight culture. The S97773 had higher OD when compared to antibiotic-resistant counterparts. So we think that plasmid uptake by these ABR antibiotic-resistant E. telluri isolate comes at a cost of host fitness. And we hypothesized that this might also come with reduced virulence when compared to the wild-type isolate. So we set up a challenge study by exposing fish to all different antibiotic-resistant isolates and the wild-type isolates. We've been monitoring survival for the last 16 days, and the wild types have reached 16% mortality with no mortality in the antibiotic resistant isolates. But this presentation was prepared 2 days ago. As of today, we have 2 mortalities in tanks challenged with the historic isolate with typical ESE lesions behind their head, which kind of is an evidence for the chronic manifestation of this disease. So moving forward, we also looked at the environmental reservoirs of antibiotic resistance in pond environment. And for that, we collected pond water samples from multiple ponds in the same farm. These ponds were separated either by tree lines or big levees so that we have like ponds from across the landscape. We cultured them on medicated plate, and we took 2 approaches. Initially, we individually picked separate colonies and characterized them by sequencing using nanopore. And in another approach, we cultured these pond water samples on different concentration of medicated plates and harvested all of the cells and characterized them by shotgun sequencing and adaptive sampling. First, I'll talk about the individual colonies that we picked. So you can see from the image, there are multiple colonies growing in the zones of inhibition around the antibiotic discs. We picked them, even though we picked more than 100 colonies, so some of them were repetitive, and these were some of the bacterial isolates that we ID'd and assembled. And as you can see, antibiotic resistance is also associated with fish non-pathogenic bacteria, which is in agreement with previous studies that show that Citrobacter species, Aeromonas, and Klebsiella species are reservoirs of antibiotic resistance in other clinical settings and livestock farms. Moving on, we wanted to look at the plasmid reservoirs associated with this bacteria that we sequenced. And as you can see, we had multiple mobile plasmids that were associated with these pathogens. And something interesting was the presence of a mobile plasmid associated with Klebsiella pneumoniae, which was previously described in Edwardsiella piscicida, evidencing towards intergeneric movement of these mobile plasmids. We also had these huge multi-drug resistant plasmid, 2 multi-drug resistant plasmid in the same Citrobacter fruentii isolate, making them these big reservoirs of mobile genetic elements and antibiotic resistance. Moving on, we also did adaptive sampling, which is basically data enrichment by targeting certain genes of interest in nanopore. And even though we targeted multiple genes for mobilization and antibiotic resistance, because I don't have a lot of time, I'm just going to focus on the genes that confer resistance to the different antibiotics that we use in catfish aquaculture. Almost all ponds had genes that cause resistance to tetracycline. Almost all ponds had genes that give resistance to fluorphenicol. And an interesting fact was that even though we did not select for resistance for sulfa drugs, we had sulfa dimethoxin resistance genes associated with most of the ponds. But this uh, study comes with some limitation. The sequencing results are not relative abundance data, they are sequence abundance data, and this may vary because of sequencing biases or genome size of the bacteria and nucleic acid extraction techniques. And the results that I've shown are based on bacteria that grew on the nutrient agar media. It's not the whole picture. There's tons of other bacteria that cannot be cultured using laboratory techniques, and to make it more clear, Phaeobacterium species, which is a major bacterial pathogen of concern in catfish aquaculture, cannot be cultured on a TSA plate. And our challenge doses did not meet the target mortality. So my future analysis will include a virulence comparison between wild type and antibiotic resistant E. coli isolates at higher challenge doses. We'll also be looking at the efficacy of live attenuated ESE vaccine against these antibiotic resistant isolates. We'll also be comparing treated versus non-treated ponds for the difference in these mobile genetic elements and the plasmid communities. So to conclude, the presence of multi-drug resistant plasmid with the genetic machinery to pick up resistant elements present in the isolates from 1994 kind of suggests that this is present naturally in the environment with or without the introduction of antibiotic selective pressure. And phenotypic analysis have also revealed the possibility of reduction in bacterial fitness with the uptake of these huge multi-drug resistant plasmids. And the reservoirs of antibiotic resistance is not just limited to the pathogens of concern, but also with fish non-pathogenic bacteria, which is similar to other, farming, livestock farming and clinical settings. This is nothing so much peculiar to just aquaculture setting. The whole idea of this research was to get a better idea about the environmental reservoirs and kind of develop better mitigation strategies. There are studies that are focusing on vaccine development and other studies that are focusing so that we do not use a lot of antibiotics. But for the time being, we need to know about these environmental reservoirs. And with that, I thank MSU CVM, my funding agencies, catfish farmers, my committee members, and everyone in the Griffin's lab for helping me with this project. And now I'll take any questions.
Great presentation. Folks, we've got time for a question if you have one. Feel free to drop it in the chat or just unmute yourself. All right. Well, Divya, you must have answered them all while you're talking. Thank you for your presentation.
Mm-hmm.